Hi, everyone. Welcome to the seminar series again for this week. And this week we have Dr. Gunawardane from Ellen Institute for Cell Science. And we're very happy to have her here. And we're excited to learn about her research on stem cells. And briefly for Dr. Gunawardane, she joined Ellen Institute in December 2020. And it's after spending 5 years at Amgen, where she worked on assay development for multiple drug targets, spanning oncology, inflammation, and cardiovascular diseases. As a group leader and project leader, she worked in multidisciplinary teams to screen novel therapeutics and drug targets. And she has also contributed to the cutting edge technologies in that company. Prior to Amgen, Ru worked at Ambit Biosciences, where she was part of a small team of scientists that identified AC220, a potent kinase inhibitor. And she received her PhD from Johns Hopkins in biology, where she studied microtubules in Drosophila and Xenopus model systems. And she combines her expertise in molecular biology, biochemistry, and cell biology. And in her current job, Ru is actually combining multiplex spatial omics, spatial RNA imaging, with a stem cell biology field to specifically target cardiomyocytes. And we're very happy to have her again. And without further ado, I'd like to turn to Ru, please.
Thank you, Ahmet and Ron, for inviting me. Can everyone see my slides? I'm not seeing anyone, so I'll take that as a yes. Okay, yes. So, okay, great, thank you. So let me, oops. All right, so I wanna kind of tell you what I'm gonna tell you about, just to kind of, have some framework for this. I want to tell you about who we are at the Allen Institute and what we do, and then talk about how we are starting to understand cell organization with live imaging. This is kind of the central focus of the Institute. And then more to what we were talking about before the meeting, I want to talk about how to use a multidimensional kind of a holistic approach to define cell states. And this is where we are trying to integrate cell organization with transcriptomics. And then I'd like to end by talking about how to build better platforms for discovery for cell biology, and of course, share that with the community like we're doing today. So I'm at the Allen Institute for Cell Science. So this is located in Seattle, Washington. We are a nonprofit research institute. Currently, we are in year 6 of the institute. We are about 70 people, highly multidisciplinary group of people, all the way from molecular biologists, imaging specialists, computational people, and also visualization and sharing. So our kind of values are team science, open science, and community empowerment. So these are things that our founder, Paul Allen, the co-founder of Microsoft, was very interested in to share with the community to help the research community. So what do we do? So this is kind of like an atlasing project. So one of our main goals, this is kind of large data-centric goal, is to create a map of stem cell states using iPSCs. And the idea here is that our focus is to do this with live imaging, because images provide a rich source of information that can complement other approaches such as proteomics or transcriptomics. And we want to use this approach to study cell behavior, such as mitosis, differentiation, and various perturbations. So really the bigger goal is to go from understand the connection from genotype to phenotype. I know we all care about that, no matter what model system we work on, but it's really hard to look at all the genes or all the proteins and interpret that information. So our approach was to kind of take a dimension reduction approach, which I'm sure you're all familiar with transcriptomic data, you do the same thing. But our approach with imaging was to consider what's happening at the cell organization level. And to do this, we are focusing on the major structures of the cell. So for example, I think all of you can look at these two images here and know what the cell is doing, whether they're dividing or not. And you're doing that because you're looking at the microtubules and you see the spindle. So that's kind of our approach, that some of these structures inside the cell, if we watch them with live imaging, can tell us a lot about the cell behavior. And by doing that and incorporating that with other data types, we think we can all better define what is a cell state and what is a cell type, instead of doing it with just one modality. And some of our bigger aspirational goals is to use this approach to really understand how cells form, how cells change, how fates are determined, and eventually how cells go wrong in disease and how we might treat those diseases. So kind of creating the new foundation of cell biology. And with the computational approaches out there and that we are also developing, we want to be able to look at a cell and be able to know what it might do in the future. So these are the predictive models we're trying to build. So for some of these reasons, we decided to use iPSCs. Number one, we want to start with normal as possible and still work in the lab. So they have to be proliferative. So unlike HeLa cells or some of the other lines, at least the iPSCs we know are diploid, they're non-transformed, and they're human. And the big other advantage is that they're pluripotent. So it allows us to watch these cells as they go about making those cell transitions and fate decisions as they become other cell types. And for today's talk, I'm going to talk about the cardiomyocyte work as well. So where do we start? So this was 5 years ago. This slide is more of a history. So we started by saying, let's map the landmarks of the cell. Let's understand where all the structures are in the cell. And believe me, I don't think any cell out there looks like these cells we see in our textbooks. We really want to know where the mitochondria, the ER, the nucleus, all these structures are, and to actually capture the variance. Because no two cells are alike, even if you look at a clonal line. So again, the approach is to do live 3D high resolution and high replicate imaging, and then developing models where we integrate all these structures into one cell and then watch the cells as they go about various cell behaviors. And you can think of your own process that you're studying, whether it's a stress response or a mutation or a disease. But mitosis and differentiation are kind of the processes we are starting with. And again, it's very important for us to understand what's the noise and what's normal variability. And that's where the scale comes into play. And then eventually what we are doing with the cardiomyocytes is to integrate transcriptomics into that picture. So we really want to build more realistic models based on actual raw data. And I'll tell you at the end how we are doing that. So again, I mentioned this in the previous slide. We want to know who is the boss of the cell, what drives organization, what drives transitions, and to do this with large sets of data where we can understand the correlations and transformations and really understand what is the code of the cell, what are the rules that drive cell organization and cell behavior and eventually cell function, and get a better understanding of mechanisms through this lens. So we started by creating this suite of FP tagged iPSC lines using a normal healthy iPSC line as a background so that we can visualize where these organelles are in a given cell. And we use CRISPR-Cas9 to do this so that we introduce a fluorescent tag at the endogenous locus of these genes. For example, to look at microtubules, we tagged alpha tubulin with a GFP. And then before I go in there, one of the main advantages there is that we are not overexpressing this fluorescently tagged protein. It's endogenously expressed because again, we really want to understand where a normal cell, where these structures are in a normal cell. Then we spend a lot of time and effort making clonal lines and doing a lot of QC, which I'll show you in the next slide, so that these lines can not only be used for our own imaging and model building, but that we can release to the community. And so we spent quite a bit of time developing our editing strategies. So remember, this was 5 years ago when CRISPR wasn't as popular as it is right now, but how to do this in a way where the cells are not perturbed. and released to the community. So I don't want to spend a lot of time on this because it is published now, but it was really important for us to know that our tagging approach was precise. It localized to the right place at the right time, and that the tagged lines, the tagged clones, were very similar in behavior in terms of mitosis, in terms of morphology, to the unedited cells. And we wanted to make sure these cells still were pluripotent and could differentiate. So we spent about 5 to 6 months generating each of our lines. We start with 100 clones and end up with the one that we share with the community. Again, we do about 17 different assays, and I just have some pictures here just to highlight some of the Western blots, the IF, we do the stem markers, and of course, once we do all this, we distribute it through Corel. So right now we have a collection of actually 43, when this slide on the left is old, 43 FP tagged isogenic iPSC lines representing, we believe, all the major structures of the cell. So whether it be endosomes, peroxisomes, adherence junctions, speckles, and of course the mitochondria, ER, all the major structures of the cell. And we do, because of this extensive QC, we are very excited about the kind of biology we're going to learn and everyone else is going to learn. So these cell lines, if you're interested, are available through Corel. And if you want to make similar lines in your own favorite line, you could do that because the plasmids are through Adgene. And of course, our methods and tutorials are available on alancell.org. So what do these cells look like? So this was kind of amazing to us 5 years ago when we looked at our first line, which was one of the first lines is what I'm showing you. This is a tubulin line. This is an iPSC colony. So iPSCs grow as kind of an epithelial sheet as a colony. So it allows us to also study some epithelial biology. And you see this beautiful mitosis happening, and it's just so clean because it's not overexpression, it's not transfection, and it's not in immunofluorescence where you have a lot of artifacts. So you kind of watch the cells as they go about dividing and all the other processes. And this is just a higher mag. This is basic confocal microscopy, not anything high res, a super res. But again, because endogenous tagging is so clean, we get a lot of information from these images. So I'll show you one more. This is a nuclear envelope line, the lamin B1 line, which is one of the most popular lines right now because you can track what's happening to the nucleus. And on the right here is a movie, and you can see two cells on the top and the bottom dividing. And this is, again, we realized the nuclear envelope actually does not completely break down during mitosis, which is not what I thought. But again, we are relearning some of the biology that might have been discovered before, but was maybe not very clear. So this is why it's just so exciting to us. So there are 43 structures. I could spend a whole hour showing you movies. But I just want to highlight that different structures in the cell do different things for different functions. So for example, the nucleolus during mitosis completely disappears and comes back, whereas the tight junctions remain intact. I'm sorry, the movie stopped playing. And they retain that cellular coherence within the colony. And on the right is our plasma membrane tag, where you can see the bottom of the cell. We just pseudo-colored in magenta. It's just exploring the cell surface. And on the top remains intact, which is colored in green. So we really do believe this approach, whether you use these lines or create them for your own work, is really a game changer because you can observe your phenotype you're interested in and get an understanding of what happens at the subcellular level in a very, very clean and robust way. So we do this with a lot of standardization in mind, and this is actually something we submitted to bioRxiv a month ago. So we use robotic automation to do the cell culture for our imaging, and we use spinning disk confocal microscopy. Again, we use automated acquisitions to collect this data. Then we use a nuclear and a plasma membrane dye so that for every cell, we know where that GFP tag structure is in reference to the nucleus and the cell membrane. This allows us to then capture where the structure is localized. And then the hardest part of this is not actually getting these pretty images I showed you in the previous slide, but to actually extract and quantify where and how much of this structure is in each cell. So again, we recently put this on bioRxiv, a paper where we looked at 25 structures over 200,000 cells, where each cell was individually segmented, both the nucleus, the cytoplasm, the plasma membrane, as well as the structure. And we've spent a couple of years, my colleagues who do the image processing, to develop the tools. Again, this is available. It's a Python-based workflow to extract, individually segment and extract each of these structures from a colony of cells. And to do this from live images and in 3D was really the challenge. So this is not 2D, this is 3D. So I just wanted to show a few of the structures here, and you can use this, it's called the Allen cell segmenter, you can find this on our website, and apply it to actually structures or proteins that look similar. It's a pattern recognition. And we use machine learning to do some of this as well. So what can you learn from doing this? So we're just starting. It took us several years just to build the tools, not just the cell lines, but the image processing tools to do this. So one of the things we have learned is that you can look at mitosis and look at the relationship between the major structures of the cell. What are the structures that change together? What are the structures that change first? And you can imagine if you add Taxol or something like that, you can understand maybe how the cell goes about making that change. And then this is our most recent work where we looked at those 25 structures I mentioned in the previous slide, where we looked at the 200,000 cells and looked at how the cell is organized, where the structures are, how much, and how variable. And I really encourage you to look at this because I don't have the time or the expertise to really describe this in detail. But the bottom line is it's not just what we see, but how do we quantify structural organization? So we're using things like spherical harmonics to parameterize the shapes, cell shape, nuclear shape, structural shape, and then understand how does that change when the cell shape changes, how does that change when another structure changes, and try to understand what are the rules of cell organization. Do the rules change when the cell shape changes? Do the rules change when the cell is perturbed in some way? Do the rules change when the cell differentiates into another cell type? So we are going to stress test those rules to try and understand how is the cell, what are the rules of cell organization, and then relate it to cell function. So that is kind of where we're heading. And that's what's described here. So what I just told you about is this focus on iPSC cell, understanding how it's organized, what are the rules that are in there that we can quantify. And then we want to apply that, since we are using iPSCs, as the cell goes about differentiation. And to get at the mechanisms of these transitions and the principles of how cells change. So next, I'll tell you our first approach to do this was to use a very different cell type, which is the cardiomyocyte. So we also have spent some time to develop robust and reproducible methods to differentiate iPSCs into cardiomyocytes for imaging. And this took a little bit of time, and we've generated cell lines now where we can watch a very important structure of the cardiomyocyte, which is the sarcomere, which is very important for its function. And by doing that, we wanted to focus in on a structure that's very important for its function and look at the heterogeneity using image-based features. But other people are using these lines to observe how the sarcomere even forms, which is really important for heart regeneration and all of that, and disease modifications, and also to follow maturation, which is a big field right now in iPSCs, to try and figure out methods to fully mature these iPSC-derived cell types, which are more fetal-like. So I'll show you a couple of examples here. So these are two cell lines that are targeting the substructure of the sarcomere with alpha-actinin-2 and titin. Titin is like the largest protein in the cell, so it was kind of cool for us to tag that. They work beautifully. So this is at day 20 and day 50, we make beating cardiomyocytes. And now we can look and watch these structures form and mature in real time. And so we wanted to explore that to do this third piece that I'm talking about here. So I just want to put that there. So I've covered who we are and what we do, and how we are approaching understanding cell organization with imaging. But now, as I mentioned in my first slide, it's not just about organization, it's about how do we build upon that foundation by using a multidimensional approach where we absolutely incorporate gene expression information. So to do that, we wanted to ask, how can we combine transcriptomics, this is where the spatial piece comes into, for mapping both transcriptomic and structural cell states so that we can define cell state even better, because we have two ways of looking at it. And even more, can we predict one from the other? Can we look at gene expression profile and predict what a sarcomere might be doing if we imaged it? One of the bigger goals is so you don't have to do 1,000 experiments to answer one question. Can we just do like 10 experiments? But to do that, first you have to generate a lot of data. And this has been submitted to bioRxiv if you want the details. So in a collaboration with Georg Selig at University of Washington, we used a split-seq approach to transcriptionally profile cells during this differentiation process. But for the purpose of this presentation, I'm not going to go into that as much, but the idea here is that we have in this boxed area 2 clusters where we have most of our cardiomyocytes from 2 time points. And the question we wanted to ask is, this is around the time point we do our imaging, can we predict what a cell might look like in terms of the sarcomere again by just knowing the gene expression profile? And the initial answer to that is no, because we just don't see enough genes changing in this time frame or to enough level if you do differential analysis that truly explain the vast heterogeneity we see in structural states. And you might not be surprised because of course there are a lot of steps between gene expression and structural organization, but we thought, wouldn't it be nice if we could? And the first part to do that is to collect these 2 pieces of information. And then of course we can fill in the gaps in between. So transcriptionally, we all have tools to do this. We can do differential expression. We can look at clusters. These are just UMAP plots. And I just want to show you there are 2 genes, MYH6, myosin heavy chain 6 and 7, that change the most when you do this differential analysis, because there's a switch between myosin heavy chain 6 and 7 as the cells mature between day 12 and day 24. We did another analysis, independent of clustering. We did a regression analysis to say, what are the genes that best predict whether the cell came from the day 12 population or the day 24 population. So this is a statistical approach. And interestingly, we found 2 genes are sufficient to 85% of the time predict which population the cell came from. So the reason I'm showing this is that it's really hard for most people to do like a spatial experiment, a FISH experiment with 200 genes or 1,000 genes. Yes, you could do things like MERFISH, but it's not straightforward. So we use this statistical approach to kind of confirm our differential expression analysis, just to say, is it possible just to use 10 genes or maybe even 2 genes to predict cell state as long as we test those in the same cells as we did the imaging. So I thought it would be interesting to show that both methods showed the same genes, and that you might not need to test a ton of genes to understand what you're looking for. But although we have these kind of tools for transcriptomics, we do not have these kind of quantitation tools to classify structural organization. So we had to build those from scratch. So here are 2 images from day 18, day 30, to similar to the time frame we did the transcriptomics. And if you look at these 2 images, I mean, we were just scratching our heads. We can't even tell the cells apart, let alone classify them in terms of structural states, right? And if you kind of zoom in and look at some of the heterogeneity, it's not even cell-to-cell heterogeneity, it's subcellular heterogeneity that we were observing. Even within one cell, we can have an area of the cell that's poorly organized in terms of the sarcomere, and I'll just say this is our actinin-2 line, which is an integral component of the sarcomere in white. So it forms like a railroad track. And so it was not just about cell classification, it was about subcellular classification of heterogeneity. So to do that, we had to use machine learning to recognize these patterns. Otherwise, there was no way a human could do this. And we categorized various patterns in alpha-actinin-2 organization. And then we tested that. So all the way from very disorganized areas in purple to very highly organized areas in yellow, which are the Z disks. And then we asked whether this made sense. So we looked at about 10,000 cells between day 18 and day 32, and each line here is a cell. And on the x-axis is the fraction of the area that belongs to each of these classes. So what we expect is going from day 18 to day 32 is that the cells would mature and would have more organized sarcomeres. This is what's known in biology. This is what we visually see. So you'd expect more yellow, and that's indeed what we see. We see a larger number of cells that have organized Z disks, as well as more cells that have larger part of their cell organized. So this is almost a gut check that our algorithm of classifying what we call local organization, because it's not cell to cell, it's more local, is accurate. And this would allow us to then scale it and quantify it. So we started there, but then we did something similar. Again, I don't have the time to get into it, for some of the other image-based features that we believed were important determinants of structural organization. And we did this by comparing it to expert annotation. So we had 2 cardiomyocyte experts who would score these cells, and we then tried to build a linear regression model, a simple linear model that used the features that an expert was looking at to create this model and to come up with this combined organizational score. So just to make sure we're not trying to come up with the best score, we're trying to come up with a score that matches the human annotation so that we can automate the process, so that we can look at thousands and thousands of cells. And so we did this, and what we are now able to do is to go from an image on the that looks like an image on the left here, which someone can look and say, oh, it's a mess, to individually segmented cells that we can assign a score to that have these 11 features incorporated into that. So if you look again to this plot here, there are certain features that are weighted more heavily. These are, again, based on the model. These are not predetermined. So yeah, the organization of the Z disk is a positive correlation, positive weight, whereas the fibers, which is very disorganized, is a negative weight, but it's all combined in the model. So we used this model and combined it. But it wasn't that easy. We had to create a workflow to do this and to combine it with transcriptomic information. So this is again detailed in our paper, but the idea is in the same cell, can we capture the structural heterogeneity all the way on the left? And can we capture the genetic or transcriptomic heterogeneity of those genes that we picked earlier on to be most highly differentially expressed during this time frame and quantify it? And we use HCR-FISH for that. And then we obviously have to segment the cells. Unfortunately, we had to do that manually, which was no fun. We're still working on how to do that in an automated fashion. And then can we look at the relationship between transcriptomics and structural organization? And we have done this now for over 10,000 cells in this time frame. So what did we find? So can we predict cell organization from gene expression? I'll give you a minute to think about that while I show you the data and before I give you the answer. So what we noticed, so on the left here, I'm showing you up that plot where we took those thousands of cells. This is actually a smaller subset of those cells that we trained the model on. So when we look at combined organizational score from left to right, the cells are getting more and more organized. And the pink cells are the older cells, the blue cells are the younger cells. And as expected, we see more of the older cells getting more organized. So on the top here is just the population analysis of that. And as expected, the older cells are more organized. And on the y-axis here, the population analysis is just showing that the gene MYH7 is more highly expressed in the older cells. This is the switch, the known biological switch I talked about earlier. So that just validated our model. And what I didn't tell you during this presentation is we spent like 2 years making this assay very robust. So the cells are very consistent, how they're made is very consistent, high QC, and the image acquisition is very consistent for these cardiomyocytes. But as you can see, the correlation is not great. Yes, there is a trend towards more organized cells having more MYH7, which is what you would expect based on just gene expression alone. But it is significant, we believe, but it is modest. And here's that plotted. So this is showing Spearman correlation between organization of the cell, which is this combined organization score, with 2 of our favorite genes. So MYH6, whether it's either time point or together, is a negative correlation, which makes sense because this gene expression goes down with differentiation. And for MYH7, there's a positive correlation because it's coming up. And we believe it's significant because we did another check with just protein, because protein to organization should be more correlated than RNA to organization, because there are too many steps in between. So this is just, that's what I said earlier, this might be a new way of thinking about it. So the idea is not to say you would necessarily be able to predict organization from gene expression alone, although that would be nice, and that was our hope, but to say that looking at both might give you a perspective you didn't have before. And we were actually surprised that we even got a 0.4 correlation. But the bigger point here is that there are cells here on the top right, 2 organized cells. Structurally, they're both organized, and this is again a structure that's important for function. But if you look, you would have expected, well, I would have expected looking at structure alone that both these cells would just have myosin heavy chain 7. And in fact, only one of them does, and the other one has the younger MYH6 transcript on. So the idea here is that you would believe MYH7, thought to be an important protein for sarcomeric organization, could be a predictor of organization, but it's not, right? There's cell context, there are all these other modifications that can happen. So it is dangerous to just look at transcriptomics and assume a structural or a functional state. So the conclusion here is that you need both measurements, even though at a population level that there are trends, at the single cell level, we do not believe you could predict one from the other. So to kind of visualize this, we've been now able to capture and quantify a very wide range of heterogeneity, just even looking at one structure in the cardiomyocyte. I'm not even talking about the Golgi or the ER or the nucleus here. I'm talking about the structure we believe is really important for its function. And then if we organize it from low to high organization, like we did with our machine learning tools, and then look at the 2 most important genes there, you see heterogeneity. You can say it goes generally from pink to green, but there are cells in here that if you try to predict one from the other, you would be absolutely wrong. So that's kind of one aspect of it. The other thing that was a surprise that we noticed is that we would expect with transcript that it could be anywhere, right? So it's first initially in the nucleus, then it's going to be in the cytoplasm. And that's kind of what we found for most of the genes that we looked at that are important for transcription, for the sarcomere regulation and maintenance. But again, we were surprised that the best predictor of state in terms of timing of cardiomyocyte formation, myosin heavy chain 7, actually appears to localize to the sarcomere. So in white, again, is the protein actinin-2, which is what we use to kind of find the sarcomere. And here in the middle, MY7 in green is the transcript. It's our HCR signal. It's not all over the cytoplasm. Most of it seems to be on the sarcomere, which is kind of biologically interesting. It might be in situ regulation translation, because cardiomyocytes don't divide very much. But we believe that this aspect, this is the spatial transcriptomic part, could be really useful in a subcellular level. We're used to thinking about it in the tissue context. Where is this cell going to be when it forms a more 3D tissue? But we believe even at the subcellular structural level, spatial transcriptomics can tell you a little bit about what the cell is thinking of doing and might do in the future, even if it's not doing right now. So we believe our map of cell states should include not just the amount of transcript in a given cell, but the localization of that transcript. So some of the things we developed as part of this study is to identify the right structure to conjoin with gene expression if you really want to make that connection from genotype to phenotype and to function. And for us, we used a prediction model or a regression model to identify those key genes that were changing in our transcriptomic analysis. Then you don't have to look at 1,000 genes. You can, of course, go back and look at more genes if you want down the road. And we had to build and develop automated classifiers of structural state, and then figure out methods on how to combine in a single cell using just imaging both transcriptomic data and structural data. And what this allowed us to do is that look at a large population of cells from different time points and be able to look at general trends, but also look at the single cell trends. And of course, we've made the code we've generated and the data accessible. Again, you can find the links on BioHub. Sorry, bioarchive, I said biohub. So we are using this approach now to study different states during differentiation. And again, our approach is structural organization, gene expression, spatial transcriptomics, and integrated and predictive models. And since I'm running low on time, I'm going to speed up a little bit. And lastly, we want to include the nucleus, a deeper dive into the nucleus to get an even better picture of genotype to phenotype. And to do this, we know there are a lot of substructure within the nucleus. That's really important for gene expression. So we want to get an integrated nucleus state to add to the cytoplasmic organizational state that I talked about at the beginning of my presentation. And we're working closely with the 4D nucleome group, so who has all these kind of very cutting edge ChIP-seq and multiple C-seq genetic information, sequencing information, so we can provide the spatial context by looking at the organization. So to do that, we are tagging about 20 different, we're creating about 20 different lines so that we can look at the largest landmarks of the cell. We've done most of these, like the lamina, the nuclear pole, the nucleolus. We're starting to do the chromatin looping, CTCF, telomeres, a replication complex, a splicing complex, etc. And the idea is to include this in our analysis of cell states and transitions going from the iPSC state to a differentiated cell state. And here's, I couldn't help it but show a few images. So our nuclear envelope, nucleolus, we have movies of this, but it would crash. So I'm just showing you stills, the splicing complex, the histones, the telomeres, etc. So if you are interested in studying what's happening inside the nucleus in your own cells, even if you're not using iPSCs, these plasmids are available for you to use. So what we are doing, kind of summarizing, is to generate a visual model of some stem cell states that include the physical organization of the cell, but where we are including things like gene expression to build integrated, holistic, but also predictive models. And to go from these kind of old-fashioned textbook ideas of what a cell looks like and how it behaves to much more realistic and real images of cells. So I want to just show you, these are two cells from a colony. One is in interphase, one is dividing, where the cells were just tubulin, microtubules were tagged. And this is where I don't have the time. We're using label-free approaches to combine and integrate different information into the same cell. So by looking at thousands and thousands of cells for each of these structures and knowing the cell shape and the nucleus shape, what we can do is know the purple is mitochondria, the blue is ER, the yellow is tubulin, the green is microtubules, the red is nucleolar. We can now integrate all that information. These are sometimes statistical models, and we have tested some of these so that we can look at the relationship of these structures in the same cell. So that's what's really exciting to us, so that we can better define signatures of what a cell type is and what a cell state is, and work towards understanding those bigger principles. So I think I kind of covered all of this. I want to end by saying all these tools, the cell lines, the plasmids, the images, the code, as well as the visualization tools, and we have a simulator, is open to everyone, and highly suggest. We want people to use it. We are only successful if people start to use it and incorporate it and study other systems. I'd highly encourage you to go to allencell.org, or of course you can contact me. We make all of our QC available to the community. We have video tutorials. We have our genomic data on our website. We have a summary of all the QC we do. The cell lines in particular are near and dear to me, are being used worldwide. And if you're part of a major university, contact me. These are our major universities using our cell lines so that individual PIs don't have to get them themselves. We're very excited to see what people find. This is a collaborator across the street who's using our cell lines to study kidney disease by making kidney organoids, but really looking at it from a very cell biological centric way to understand fundamental mechanisms that go wrong in kidney disease. But this is similar to what a lot of people are doing right now. So we have a lot of different data types depending on your interest. Also a lot of educational materials for teaching where you can kind of interact with the data. So I want to end by saying thank you. This does take a village. There's only about 20 experimentalists, but the rest of the people support the work, computational people. It does take a village to do this. And I want to end by thanking our amazing and visionary founder, the late Paul G. Allen, for his encouragement and support. So with that, I'll stop. Happy to take questions, and I will stop sharing.
Thank you, Ru, for this great talk. Any questions? There's actually a question in the chat, Ru. Can you read it, or should I help you read?
Oh, yeah, I can open the chat.
Mm-hmm.
Yes. So the question is if you have a multi-layer organoid. We are testing this right now. We're taking the iPSCs through the early transition, the what we call EMT, the epithelial to mesenchymal transition, and it forms a layer. It's actually forming a tissue. So our idea is to apply those principles, test those principles during those transitions. What we've found, and you'll see this in the iPSC variance paper, is that the relationship where the structures are very consistent. We're very surprised, very robust across the cell shape space. So there are like 250 dimensions of cell shapes. And the localization is very consistent. The main variables we found in epithelial cells is that if you do a PCA analysis, is cell height and cell volume. So if those two things are changing, the cell's shape will change. But where the structures are localized, don't seem to be changing very much. So. But the challenge, I think, is how to measure that, right? And that's kind of what we hope people try and use what we did and apply to their system.
Yeah, I think the response in the chat is very cool. So I have a question. Since I'm a biomedical engineer, I think often after my colleagues studying kind of cardio tissue engineering, they're really looking at how the cardiac cells, beat the mechanical property. Have you kind of ever look at the rate of the cell beating? And also, if possible, you can measure the force generated by individual cells. How does that function correlates with the structure of the sarcomere you measure?
Yeah, that's really important. So yeah, we're doing some traction force microscopy with the collaborators. So we try to think of things that other people are not doing and to do it at scale. So how we are right now operating is we're collaborating with the cardiac community who are looking at some of these mechano assays, like Beth Pruitt at UCSB and several people at Stanford. So they take our cells and do those measurements, and we are focusing on kind of the organization piece. We actually have to stop the cells from beating sometimes to image it because they move too much. Then you can extract and segment the cells. It kind of defeats the purpose. So I think we have to do things in parallel and then try to integrate. That's where the integrative approach is very useful. And for functional assays, you can do 10,000 cells, right? So that's the challenge. The scale is sometimes a challenge, but very important. We are hoping to combine those data with our collaborators.
Yeah, I think you mentioned ideally you can measure the spatial distribution of all different transcripts and kind of correlate with the sarcomere structure or whatever kind of subcellular structure you are looking at. So I know in neurons that makes sense. People found the transcripts, different regions of the axon, actually highly specific. They call that kind of RNA zip codes. But yeah, I haven't heard about that in other cell types. I'm curious, if you can educate us a little bit. If anything, people learn in this field, sort of the cardiomyocytes, the transcript distribution that's correlated with kind of regional translational activity.
Yeah, so when we first saw this, we were like, has anybody else seen this? We found a zebrafish study that also saw this transcript localization at the organelle. And we have heard that it's just more for regulation. There's a ton of transcript there, and then it's more about stability in terms of then it gets translated. So I think this is a different level of regulation because the cells are not proliferative. So they have to have like this huge burst of transcription, and they seem to be localizing to the structure. So of course we haven't studied this. This is just speculation based on discussions with the community. So there are some proteins, I can't think of the name, that are more like ubiquitin pathway, also localizing to this structure. So there's some kind of a post-translational and post-transcriptional regulation going on at the structure. But how it gets there and stays there, I don't know. It's, I think, very cool.
Data.
Yeah. So I want other people to start looking. I don't know how common a phenomena this is other than the neurons you described and the zebrafish study, which was also hot.
Yeah, this is super fascinating. So I think that becomes not just a kind of local translation activity, but it's a kind of equilibrium. You have more transcripts there, and it's super cool. I think it relates to, I think, another concept I think very intriguing in the entire field of cell biology is the phase separation and local condensation.
Yes.
That might be related. Yeah.
Absolutely. We are seeing some very interesting phenotypes with speckles, which is where the transcription, the splicing happens, and heterochromatin, because we have now all these structures. We're like, oh. So I can't wait for us to analyze that data, but I think there's just so much we don't know, right? And we make assumptions by amount of a protein or amount of a transcript. I think we really need to understand where is it? And based on where it is it in the structure that we thought takes another 20 days to form, is it already there seeding the structure when we usually think it's a protein? I think the fundamental understanding of how the cell forms, how these structures form by looking at a transcript and looking at a structure, to me, I am relearning cell biology.
Yeah.
And then I want to mess it up. That's why we want to do perturbation. So we have CRISPR-I screens and mutations, but we are so still fascinated by what we are finding with just the normal cell and so much variance.
Yeah.
And then one question also from here. Oh, there's actually a question also on the chat. After that.
Yeah, from Joy. Actually, you can also unmute yourself, Joy, if you want to talk to the speaker.
We have not done a mixing yet. We are starting to make some endothelial cells and want to mix it in and see how they relate.
I'm really kind of curious how they kind of communicate with each other. Can you capture that actually as well with these methods and technology? And especially in forms like for kind of neurons, how do they interact with each other? But could you also kind of visualize interacting neurons with astrocytes or and so on? I'm kind of curious.
Yeah, there's no reason we wouldn't be able to. We would have, as long as we can segment the cell and as long as we know which cell is which. So the first thing we actually working, the Allen Institute has Allen Institute of Brain Science, which you guys might know a lot more about. They are the original Allen Institute. So we're starting to talk to them about that. So we haven't done it yet, but we would like to. I do want to say, even without mixing cells, we do see the context of the cell within the colony has a lot of influence on cell shape, cell height, the y-axis, the z-axis. But we also see with the cardiomyocytes, this thing where the cells eat one another. It's very disturbing to watch. But they just go and engulf another cell. So we saw a lot of multiploid cells and we just assumed it was because of cytokinesis gone wrong. But then when you do the dynamics, when you make a movie, we see it's not the cytokinesis. There's definitely some cells that are not cytokinesing, but they engulf each other, and it's very specific. They will go over one cell and do it to another one. So it's not just the neighbors. So there must be some local environment they're sensing. So yes, we're very excited about cell-cell interactions. It's a little bit more complicated because you have to track the different cells in time. But we're happy for others to do it and use our tools. We can't do everything.
I can kind of think about if you can have like the structures for neurons and astrocytes like separately build it in your model and in your structures, that you later can translate that to mixed cultures, that you kind of deconvolve your mixed cultures based on the structural data that you already have.
Yes, which is exactly what we did to put together the different structures too, right? Yeah, exactly. Basically what it comes down to is having a really good training set. And that is a consistent, robust, and large numbers. And then you can scale back. The whole idea is dimension reduction, but to do that, you have to have a lot of data to start with. That's really good, high quality.
Thank you.
You're welcome. Thank you for the great question.
And then one question from here. So when you mentioned that you just can use 2 genes and 5 genes and 10 genes instead of 100, right? I think this is very important as well, especially if you want to understand this differentiation and others. So my question is the technology differences, right? You use FISH image-based, the efficiency is high, and other sequencing-based technologies, the efficiency may not be as high, right? And when you try to combine these data sets, would you observe some technical issues because of the efficiencies?
Yeah, just to align the cells. So we first live image the structure, then we fix it and do HCR and come back and image it again. So when you are talking about cells that are in a population, to know where that cell is, even though you are fixing the cell, there's some movement. So there's alignment, right? And then the other big issue is how to tell one cell apart from the other cell when a lot of cells are kind of stuck together or a little bit on top of each other. So those were where we found the most challenges. We did want to do about 10 to 15 genes in this at the same time. So we did try RNA-scope, didn't work very well for us. There's just too much background. Again, high quality is very important if you're going to spend the time to do all this type of image processing, right? And machine learning. The data quality is really, really important. So, HCR worked really beautifully for us to do 2 to 3 genes at a time. And of course, because it's live endogenous tagging, structures are very clean. Then it was more of a technical thing, and segmentation was the hardest thing because there are no really good dyes that go to the cell membrane and stay in the cell membrane, right? Because there's endocytosis. So that's a technical challenge.
And then do you also try to align your data with the RNA-seq, the regular RNA-seq as well? Do you compare them?
Yes, we didn't do it for this study because the RNA-seq happened like a year before we did all the imaging. But in the future studies we are doing right now, we are doing a parallel analysis where we are imaging and we are also doing RNA-seq. And we're going to pick our genes based on that so that it's really done with the same cells.
Because like to reduce the number of genes, like in an imaging experiment, actually you can use the data from the sequencing, right? And
yes, I mean, that is how we did it for this study too, but we always were not sure because it wasn't the same cells, right? And the time points were slightly different because imaging requires more time for the cells to be happy on that different platform. Yeah.
Right. Any other questions?
So actually, recently we figured so actually if you can image your cells and cultural cells on the substrate, so we can sequence exactly the same cell you imaged. Yeah.
Yeah, exactly. The difficulty with that is the cells, there are numbers of cells.
That's right. Yeah.
Yeah, there's always some kind of challenge. But.
Yeah, so I just kind of posted in the chat. I noticed quite a lot of people are sharing the same Zoom link. Yeah, please don't do that because I may get another email from Zoom telling me there's a kind of security concern. So just go ahead and register your own Zoom link. Yeah, you get your own link almost right away. So thank you very much.
Mm-hmm. And thank you again, Ru, for this fantastic talk. I think it's kind of clapping time for you. Appreciate it.
I'm gonna do the real clapping.
Thank you for inviting me. It's been a pleasure, and I'm looking forward to attending this in the future.
Please do so. And before we conclude, I'd like to also mention that next week we will be hosting Dr. Bo Huang from UCSF, another imaging expert. And he will be talking about mapping the inner world of living cells with pretty much also subcellular movies and cool data sets. And we look forward to seeing you guys next week. And for now, have a nice weekend.
Thank you all. Bye.
Bye.